Good morning, students. In previous class, we started discussing about clinical trials, right? We have completed the introduction to it, the types of clinical trials, and a general introduction to phases of clinical trials. So now we are going to discuss in detail about phase one clinical trials. Okay. We have seen definitions. We have seen development of clinical research, and why it has been happened. Good development after Second World War. We have seen the reasons. What are the basic principles? Your drug should fit into one of these parameters to be a new drug or to do a clinical trials in it. Okay. And next, yeah, we've seen the types of clinical trials. Like this must be the basic the principal consideration requirements. If whenever you're going to do clinical trials, factors to be considered before designing a clinical trials design. Next, yeah, we have seen the types like treatment trials, prevention trials, diagnostic trials, screening trials, prophylactic trials, quality of life trials, and this is a overall thing about your clinical trial phases we have discussed already. Yeah. Next, we have seen this also. We have seen this. We have completed this. Yes, we we were here. So phase one clinical trials. So in phase one of clinical trials, we have discussed about what we are going to do in phase one clinical trials and the objectives of it, like safety dose, tolerability, PK and PD of the drug, and prerequisites for phase one. We have seen conduct of clinical trials. We have seen and players of clinical trials. We have seen players are nothing but your stakeholders, like stakeholders and people who are involved in the clinical trials. Next, we have seen definition of healthy subject, right? And next, we are going to start with this. Yes. So I was being saying healthy volunteer studies. So how come healthy volunteer studies are well understood and can act as a reference point to gain an understanding of a test compound? Means so here they're saying how better your healthy volunteer studies are good and how can it will act as a reference point. See here why I'm saying how we are saying why we are saying reference point is whenever you want to see these objectives of phase one like safe dose, PKPD and all. So what happens here? We want to know as a patient is healthy, he don't have any specific disease conditions which can bias our clinical research or phase of clinical trials. So we'll be seeing the basic parameters like what is safe dose, what is the tolerable dose in a healthy volunteer, where his disease which is not involving or interfering and leading to bias of our products, bias of our subject, right? So here we are going to see. So first thing is involving healthy volunteer in these phases may be advantages. So obviously it will be an advantages. Like it will we'll be getting the precise results of your safety, tolerability, and PKPD. Because whenever we involve any patient whose disease or patient who is having disease condition can interfere with our clinical trials, which can lead to bias. So that thing won't be there. So it will be precise. We'll get the quick data and it will be low cost. So why we are saying low cost means supposing if patient is having a disease condition, we should take certain extra precautions for the patients while giving the dose or while checking the parameters. So we are going to leave all those things. We are just going to go for just a minimal procedures which we are going to do when we are involving healthy volunteers. So that will leads to low cost for the sponsors. Next, more accessible and physiology is well understood. So why we are saying more accessible and physiology is well understood and control over the right means. So here what happens? So we already know what is the physiology of healthy person as being increased in the technology and with the knowledge we can understand. Okay, once you take a food, how it's going to digest. how your immune system is going to work, how your immune system is going to work. So we already know all these things, right? But when you see a diseased person, we don't know he is in which stage of disease condition, right? Even if we take him to see the safety dose in him or on what happens, the disease it won't be at the same stage, ma. It will be changing its state, maybe from stage 1 to stage 2 or stage 2 to stage 3. So it will be a problem. So we have to see the physiology of the person each and every time. So that will leads to heavy, and it will be a tedious or laborious process, and it will be cost increasing process also. So there are a number of multiple factors which are involved in this problem. So it will be better to use a healthy volunteer in phase 1. Right, next, no disease condition, no medication taken, no special monitoring isolation, like we are discussing in this point. So if patient don't have any disease condition, we don't need to take any extra precautions to say to see the results in the patient, right? Your PK/PD values are safe to tolerable dose. So we'll be completing the thing a very early, and we'll be completing with a low cost, right? Next one, dropout rate will be less. So dropout rate is nothing but people eliminated from studies. So generally in dropouts, there will be many reasons, maybe minima, like patient don't want to be part of it, and our patient is no longer required for the study. Okay, patient disease conditions is being increased, or patient have a new disease condition which will interfere with our studies, right? So whenever we cannot continue the people in the study, so we have to eliminate them, we have to remove them from the study. That is nothing but your dropout rate. So consider you have started with 100 people, and after completing your phase 1, or after completing your studies, only 70 people are there with you. 70 people were there with you. Remaining 30% are dropped out, means they eliminated or removed from the study, right? So that is dropout rate. So out of 100, 30 people are eliminated or removed from the study by the end of the study. Okay, you started with 100 people, and 30 people are removed or eliminated before completing the trials. By the end of the study, you have only 70 people with you, means 30% is the dropout rate. So this one will be less in case of healthy volunteers, as they don't have any disease conditions and all. If it is a diseased people, like I said, the disease condition may be progressing, and maybe certain comorbids also happen to the patient. Like if it is diabetic patient, long-term complications, definitely retinopathy or neuropathy, diabetic retinopathy or diabetic neuropathy will be there. So that leads to a problem for the clinical trials, right? So at that time we have to eliminate the people from the study. We have to remove the people from the study. So this kind of things won't happen when you take in healthy volunteers in the study. Next. So like we said, like when you see the healthy volunteer definition, it includes what it says in WHO. Patients whose disease condition won't interfere with our study, we can consider them as a healthy volunteers. That's what they said here. Although not normal practice, individual with mild but stable illness such as hypertension or arthritis could be considered as a phase 1, considering US FDA definition of a healthy volunteer or normal subject. Right? Like hypertension, arthritis, and nowadays it's becoming a common this thing. So if your drug is not going to treat your hypertension, arthritis, it is for anti-epileptic or some other thing. Yes, you can take the people with chronic or normal conditions like disease conditions like mild disease condition like hypertension, arthritis. Here, this is what we have seen. According to FDA definition, subjects or volunteers with a mild stable illness are considered healthy if they don't have diseases for which drug is being tested, for which the drug is being tested. It means if patient is having hypertension, arthritis, and you are dealing with anti-epileptic drug or antibiotic, then obviously they don't have any link with this one. Means antibiotic is not going to treat hypertension or arthritis, obviously, right? So you can take those kind of people in the study, and existing does not complicate the interpretation of the data. It means because of his disease conditions, our clinical trial phases should not be affected. They should not be biased or confounding. They should not. his disease condition or her disease condition should not lead to wrong interpretation, should not lead to wrong interpretations, right? So that's what the definition of FDA, and previously we have seen the definition of it, obviously, right? Okay. This is just leave it. Yeah. So like we already been discussing, we don't include healthy volunteers in any trials which we are dealing with HIV or cancer, right? So see, the same thing says here. For certain disease conditions like HIV and cancer, real patients who have end-stage disease conditions and lack of other treatment options can be. It's just it can means you can or there is it's not definitely you can include in phase one trials because we don't include people in this trial because we already know the drugs for HIV and cancer is having much severe side effects. We are not going to use healthy volunteers in phase one trials. Okay, same thing. See, for oncology or HIV trials, inclusion of healthy volunteers is ethically not acceptable. Like when you see the normal guidelines, you have to take healthy volunteers. But when you see ethically, where we are going to, uh, where we should not expose the people to unnecessary risk, so we should not include because we already know there are no toxic effects for these drugs. Like we seen, the effects of cancer already, anti-cancer drugs, like nausea, vomiting, alopecia, and some other thing. So many things here as we already know. The side effects of anti-cancer drugs are so severe, right? So we cannot include. So instead, we'll be taking the people who are at any stage of disease conditions, or we'll be taking a diseased patients. We may not be at any stage, people who are at early stages or in anything. We can just take a few people, like 10 people maximum or 20 people maximum. We can conclude, we can get some PKPD values because we already have certain data from the animal studies and we are going to extrapolate the data to human studies and we are going to check in there, right? Next, procedure for connecting clinical trials. Right now, phase one clinical trials. First one, subject recruitment. How we are going to recruit the subject? What are the precautions you are going to follow, and what are the criteria you are going to use for selecting a subject? Next, informed consent. So, informed consent is nothing but where you are going to explain to the people what you are going to do and taking their consent. Screening means you are going to screen the patients before you will start with clinical trials. Because if we say I'm healthy volunteer, we cannot believe in him, whoever it may be. We just have to go to certain laboratory parameters and then we have to decide whether he's a healthy person or not. Next, recording baseline parameters means before we give a drug, what is his creatinine clearance, renal excretion, blood pressure, all the vitals, all what is the kidney function, what is the liver function, what is renal function, everything will be checked. Next, drug administration means after taking all the basic parameters, you are going to administer the drug to the patient. Okay. Next, you are going to collect the blood samples of the patient. Next, record the post-treatment parameters. Means here you are going to record the post-treatment parameters. How it is. Is there any change in the creatinine clearance or renal excretion? Supposing the patient's creatinine clearance is 1.5, and after giving a drug it was 1.8, right? So those kind of recordings you are going to check. Next, analysis of blood samples for a drug level means whichever whatever blood samples we'll collect from the patient. So from that blood sample, we are going to see what is the Cmax, Tmax, and all. Means how much drug we required. The patient, the drug in the patient's body or healthy volunteer body should reach the Cmax and Tmax. And we are going to see for specifically when you see in phase one, it's specifically about a safety, right? Safety dose and tolerable dose. We are going to see those two things also, and PKPD parameters will be checking in the sample. This is just a brief about how we are going to connect phase one trials. Next, data collection, data analysis, statistical analysis, and report generation. So after getting the data, you are going to analyze the data, you are going to generate a report. of your procedures and changing the drug and blood, changing the blood samples. Like whenever you see here, we said basic record baseline parameter. So same parameters you're going to check and you're going to see the difference in it. And you're going to find out the difference is because of the drug or for some other reason. To find out whether the changes is because of the drug or some other reason, we need to have two groups like control group and standard group. Okay. Design of phase one, like I said, to determine the variation, like we discussed in previous slide, to see the changes in the blood parameters, some other thing, or because of the drug or because of some other group, right? So that's why we should have a placebo as a control group. Like we have, like I said, two groups will be there: treatment group and control group. So in treatment group, you are going to give your drug, and in control group, you are going to give placebo, right? So we see in control group, it's not that you are going to give only placebo. You can give any existing treatment for the disease if they have. Okay. So in types of control, so one type of this one is you can use placebo as a control to determine the variation in adverse drug event. See, not only just adverse drug events, it may be any changes if you want to find out. Supposing you are given a drug, you are drug to the patient, and there are certain changes. That changes are because of the drug or because of some other environment factors or some other thing that we have to check. So to see that one, we should have a control group and test group. Okay, so in test group you are going to give your drug, and in control group you are going to give patients. Sorry, in control group you are going to give any placebo, where there is a nil therapeutic effect, right? So now we are going to see the changes in the baseline parameters or changes in anything. If any ED happened or anything happened, any test dose or all this thing you'll be seeing, and you'll be comparing. Suppose consider in phase one, you are going to check with the drug, where in test group where you're giving a drug, there are certain side effects or adverse effects, and in control group there are no side effects or adverse effects. Now you can directly say the drug is having adverse effects. Instead of it, after giving a drug, if there are certain changes in the laboratory parameters, if the changes are similar in both control group and test group, so what happens, this changes is not because of the drug. Let me give an example here. Like when you consider there is an increase in urine frequency rate, there is an increase in urine frequency rate, or there is an increase in GFR, there is an increase in GFR. If the increase in GFR is same in both control group and test group, then the increase in GFR is not because of the drug. That one is a normal psychological effect. Even psychological effects plays role here sometime. It's not like that. If you see here, here the laboratory values, the change in laboratory values are due to investigational agent or any other factors include like a psychology. See, many subjects feel they're taking medication, they should have certain adverse effects or this kind of thing. See, the adverse effects can be pain, which we cannot see the pain, right? We cannot see the pain. We can just have to feel it when they say they have pain. Or if someone said they're supposing if they say the temperature increase, you can just check it. If the fever is ADR, you can just check it. But if it is pain or certain stomach upset or certain gastric burning, that we cannot see. So sometimes what happens, people will have a psychology intention, okay, we are taking a drug, we should have certain symptoms. So these kind of things will be there, right? If someone says a person who is taking a test drug, means active drug, and if he say he's having certain symptoms of gastric burning or stomach upset, so how can we check it? We just have to know, okay, this we just have to note down, okay, patient is having upset when you take these medicines, right? Okay, and that to the problem is this ADRs may not be happening all the patient as you know it. Okay, so this kind of problems will be there. So if the same thing happens with a control group also, if control group a person who is taking control thing means a placebo, if he also says he's feeling some upset or gastric burning and all, then obviously it is certain other environment factor or psychological factor, a change in any of this light temperature diet or caffeine, taking caffeine or diet or all those kind of factors. That's not because of the drug. So that's why we should have treatment group and control group. And there's certain common placebo factors like headache, dry throat, lethargy. Everything will be there where we are going to see the symptoms are happening in test group and control group and what is the difference between these two. Okay. Next one is design of phase 1 studies. Subject selection based upon the criteria. Based upon the criteria. Like if you say in your inclusion criteria and exclusion criteria. So if you say in inclusion criteria, you're going to take people into a study whose age is equal to greater than 18. So you're going to take only people whose age is equal to or greater than means 18 or greater than 18. And exclusion criteria will be like if your person's age is less than 18, then you are not going to take them. So like this, you will be having a criteria. So based upon the criteria, you're going to select the subjects to your study. Next one is randomization and blinding. So this one, like randomization blending, I already know it. You are going to select a group of people. Then randomly you are going to divide the people for experimental group and control group, to which you are going to give experimental treatment. The other you are going to give a standard treatment. Means if there is a treatment already existing for the disease condition, we are going to check with this. And blinding means you are not going to say to the people whether you're going to give test drug or standard drug. Means placebo you are giving or a drug you are giving treatment. Like next types of trial design. So that trial designs may be parallel or crossover. So parallel means like this. So one group will receive only experimental, one group only standard treatment. So crossover means after some days, the control group will receive the experimental drug and the test group will receive the placebo, right? So mostly the crossover designs we don't follow crossover designs because once you give this drug and you want to shift the people from receiving experimental test drug to placebo, a standard treatment, what actually happens here is that these people has to wait for a certain period of time until your experimental drug will leave the body completely. Means that one is called as Windows period or washout period. Washout period means the drug should completely clear from the body and there should not be any effect. 0.05% of effects also should not be there in that person. So after making the person the person clear, then only we can give that person a standard treatment. So it is time being, it will take a lot of time to wait if a person has to complete wait for a washout period or this one. It may take 7 days or 10 days, really more time also. So that's a problem. So we don't check with it generally. Okay. That's why your parallel is the crossover design is not much welcomed in the clinical trials. Next, dosing estimated for safe dose of volunteers. So dosing estimated for safety of volunteers. All the available information to be considered. Like generally when we see when we go for this phase 1, we have certain data from preclinical trials. So that data should be considered and we should go for doses acceleration. Like estimation of the first dose in humans. This is the first time we are going to give dose in humans. So calculated based upon the data obtained from the animal studies. Yeah, obviously, if in animal studies, if they say 500 mg of 500 mg of paracetamol is safe dose in animals, so obviously what we'll do, we'll extrapolate the thing. We will extrapolate the thing that 500 to 650 mg. So we'll use first 650 mg and we will see. Okay. Then we'll use 750. There is no problem. Then we'll use 850. Then we'll use 1 gram and we'll see. So like that, we'll extrapolate the dose from animal to human. Like we have seen Noel approach and another one is Mabel approach. So what is this Noel? So Noel, the full form of this one is no observed adverse effect levels. Means the highest dose of a drug without any sorry, which won't produce significant increase in adverse effects and are relevant doses in adjusted by the appropriate factors, right? So this is the highest dose where it won't produce any of adverse effects. Suppose think 4 grams of paracetamol is a normal dose per day, right? So above which it's going to certain side effects or adverse effects. So that is a novel dose, means no observed adverse effects, means at that level they are not observing any adverse effects, right? And next one is MABEL approach. So MABEL approach is for high risk medicine generally. So MABEL means minimal anticipated biological adverse effects. What is that? Minimal anticipated biological effect levels. Okay. So here we are going to see an anticipated dose causing minimal biological effects in human approach. Okay. So we'll discuss in next slide in detail about this. What is this MABEL approach, right? So MABEL approach, the calculation of yeah. So when you see the calculations of MABEL approach, you are going to utilize the entire data whichever we got it till now. We got it till now in animal studies or human studies. Okay, like in vitro, in vivo studies, like receptor binding studies and receptor occupancy studies. Means what is the effect or effectiveness of the drug, means how much a drug is going and binding to the uh, on target receptors and how much drug is going and binding to the off target receptors, and what is the receptors occupancy rate in vitro and in vivo, whether in human studies or animal studies we are going to take it. Next is the concentrations curves like Cmax, Tmax, and steady state concentration things should be taken care. Next, exposure at pharmacological doses in relevant species. So that one also should be considered. And these up two approaches will be using to give a first dose in humans. Which what is the first dose we can give in humans, right? And whenever you see this MABEL and novel approach, so this novel approach is for normal all the drugs, and this MABEL approach is for specifically high level drugs like antibiotics or your narrow therapeutic index drugs. So those kind of things, right? Next one, dose escalation scheme. So in dose escalation scheme, we are going to see the range. We are going to see the range of the means what can be the minimum dose we can start with in humans. Like, so whenever you see these lines, they said the tested dose will usually be fraction of the doses shows harm in animal studies. Like we said, supposing 500 mg of paracetamol at a dose of 500 mg of paracetamol animals, if they started showing adverse effects by 500 mg, okay, so we are going to start with a 500 mg because as based upon the body surface area and the systems also adverse effects will vary. That's what they give. The tested dose will usually be the fraction of doses that show harm. So 500 mg has shown a harm in animal studies, in animals like in rodents and non-rodents. So we started with just that dose in humans. So that's how we will start the dose range. We'll start with 500 mg. We'll see is there any tolerability levels or toxic levels, or whether 500 mg is safe. So we came to know okay, 500 mg of paracetamol is safe in humans at a time, right? So for doing that one, we'll choose two studies, single ascending studies and multiple ascending dose ranging studies. So we'll start with the single ascending dose studies now. So what is this single ascending dose studies? Okay. So this single ascending dose studies, what they will do. So the term itself indicates single ascending means they're going to give a single drug at a time, and they're going to see it. Like let us discuss in detail about this. They're going to take a small group of people. They'll be given a single dose, like they'll just do 500 mg, and they'll just observe it. So is there any toxic levels, any ADRs or anything has happened? They'll observe it. Okay. So when they do this one, the considered dose is 1 to 2% of maximum tolerable dose in animals. So we had certain safe dose in animals. We have certain tolerable dose in animals, right? So that's what we are going to check. At this point, says considered safe is 1 to 2% of maximum tolerable dose in animals. Means how much the tolerable dose in animals has shown. That may 1 to 2% of this animals humans dose and all, right? Next one, when we are performing this single ascending dose studies, we are going to see this. We are going to follow these steps now. First step is subject selection by criteria. We already seen this. Next, when we are seen, when we are taking the people in phase one, we are going to hospitalize the person. Means that he should be in your area, or your premises where you are conducting the study. You will take urine samples, blood samples, and analyze it. Okay. After that, what you give a drug. You give a drug to the patient. After giving drug, every 4 hours, your blood sample, urine sample should be analyzed. Every 4 hours, your blood and urine sample should be analyzed to see any physiological changes or to see any thing in the blood and urine. Okay, it should be changed. Next one, and every 4 to 16 hours at initial times, you should check the patient ECG also. Okay, is there any increase or decrease or any problem? Is there like a PQRS tears or anything? That one should be cross-checked again, right? Next one is final sample. Means after whenever you see this every 4 hours you'll go for blood and urine sample, and within 4 to 6 hours you check for ECG. The final sample says within 24 to 48 hours after dosing, means after giving a final dose to the patient, that one where you will be checking about 20 or 48 hours at maximum. Means by 48 hours you are expecting by 48 hours the drug will lose its efficacy in the body. Means it has eliminated and there is no effects in the body of that drugs. Okay. And before discharging, before you discharge the subject, you should examine the subject, all the parameters which we are being saying here: ECG, blood, urine, kidney, renal, and all those things. Okay. And after even discharging, you should follow the patient up to 4 to 7 days to see any post-treatment effects or post-drug administration effects. You should follow the patient for 4 to 7 days. Okay, because some areas will be like the delayed type. We already seen some areas or certain effects will be delayed type and done. Okay, okay, and if no adverse effects found, then the next group will start with a higher dose. So here they started with 500 mg of paracetamol. They're given all the things, and they found there is no problem. So next batch they will receive 650 mg or 750 mg. Okay, and they follow all the approach, all the steps. And if there are no problems seen, then what they will do, again they will the third batch they will start with the 1 gram. Okay. So like that they will repeat the thing. Okay, so this process continues until in which batch, okay, the pre-calculated PK safety levels reach are intolerable and side effects shown. So yeah, this is the major objective of your phase one, right? To see the tolerable dose and to see the side of safety dose and PK/PD, right? So with the help of this softwares and all, we can just continue to have a pre-calculated PK safety levels in software. So till the time we reach the maximum Cmax and maximum Cmax, or area under curve, or interval dose, or any side effects shown by the drug. So we'll be repeating the thing. Your single ascending dose will start with 500 mg, second batch 750, next 1 gram, next 1.5, next 2, next 2.5, next 3, next 4, like this. We'll be seeing until our pre-calculated PK safety levels have been reached, and we are seeing certain side effects there. Okay. So once we got certain data from single ascending dose, okay, we came to know, okay, 500 mg. In this slide when we were discussing, I said 500 mg and 50 mg, 100 mg like that. Sorry, 1000 mg like that, right? Or 1 gram. So we came to know 500 mg is safe, or 1 gram is safe, 1 gram of paracetamol is safe for one time, right? So that is single ascending dose we came to know. So what we'll do with the help of this single ascending dose data, we'll go with multiple ascending dose now, right? So after we got the results from single ascending dose studies, okay, then we are going to start with it. Means your multiple ascending dose studies is as similar as, means the process is same for multiple ascending dose studies also. Like you'll be giving a drug, you will see in one batch. If there is no problem, you will start with the second batch with higher dose. You will see that batch. If there is no problem, you will start with third batch with higher dose, like that. We'll follow the same process like what you have followed for single ascending dose. You'll administer the dose, you'll see the basic parameters. Okay, it will be done to see the PKPD parameters, but this is for multiple dose, means you'll use 500 mg at the 650 mg in morning, and you'll use 650 mg in the afternoon, and you'll use 650 mg in the evening. And you see there is any problem, and this one is to see the half-life of the drugs generally, right? You will see there is there any problem, no. And what you will do, you will start giving 700 mg or 750 mg morning, afternoon, night. And if you see any problem at the 700 mg, then what you will do, you are going to stop there, and you are going to make that one as a standard dose, and same thing you'll repeat in next batch to confirm whether that is safe, same or not. Okay, so like that, what you are going to do, you are going to give the multiple doses and see the results in it. Okay, so in here, what you are going to do, you are going to collect the samples at different points of time, and you are going to analyze how drug acts in the body, like you're nothing but your ADME. Okay, supposing, like I said, 700 mg or 1 gram was safe at one time in a patient, if you consider that one. So that is a maximum dose we can give to one patient. But to reach from 500 mg to 1 gram of paracetamol safe in, per people for one time, who don't have any liver problem, is we have to go for multiple doses and multiple batches. Supposing batch 1 receives 500 mg 3 times, batch 2 receives 650 mg, batch 3 receives 1000 mg. So at a point of 1000, you came to know there are certain side effects, and that is a tolerable dose. So what you will do, you'll be having a batch 4 where you will be seeing a 900 mg or 950 mg, because at 1000 mg it's showing side effects. So we'll reduce the dose and see at 900 mg is there any side effects or not. If there is no problems at 900 mg, you will start with a fifth group, 950 mg. So in 950 mg, you will be seeing it. Is there any problem or not? And if you find any problem in 950 mg, like that is a tolerable dose or side effects is there, then you will stop there, because you came to know 950 mg is the maximum dose of paracetamol or certain next drug we can give to patient in a one time. In a one time, and that leads to multiple doses, 950, 950, 950 are given. It's almost 18.5 grams in a day, right? So that is almost safe in patients having liver problem. So like this, you are going to increase the dose and decrease the dose and see in each passage in clinical trials. Okay, and next thing we are going to see is pharmacokinetic studies, like your ADME. So generally, when you see this, to establish these studies, we are going to see to establish therapeutic schedule and to adjust the dose for drug therapy monitoring, like generally for narrow therapeutic index drugs. And we are going to see the thing, how the body will act in the, sorry, how the drug will act in the body. Okay, and sometimes what happens, we cannot go for PK studies, because if we know the drug is being a risk to a patient, that time it will be the parameters will vary, and based upon each type of clinical trial, like we are going to do radiation or a drug or a vaccine, they are going to change it. Okay, we are going to see the ADMA of the drug, and plasma profile. We are going to see Cmax, Tmax, AUC, everything will be calculated here. Next one is the elimination rate specifically should be checked in healthy volunteer to see how drug will act in healthy volunteers. Okay. And next, these pharmacodynamic properties are not expected to change, ma. If in your healthy volunteer elimination rate or pharmacokinetic, if you find the half-life of the drug is 6 hours, then the half-life of the drug should be 6 hours in all the people. Because why, if the half-life or elimination rate is increased or decreased in some other people, supposing for now we know we can give 650 mg of paracetamol per day. Okay, so in clinical trials, if we came to know 650 mg is normal for every 5 hours, and if we give the same drug to all the people, if the elimination rate or half-life is not 6 hours, or if it is 8 hours in some other people, what happens? It leads to drug deposition, it leads to toxicity, right? So if in case there is changes in any kind of people, so that changes should be noted and find out why there is a change. Is it because of the person's different physiology, or is it because of patients having any unknown disease condition which interfering with our studies? That should be taken care. Next one is your pharmacodynamics. So this one we are going to see to find out the dose response related to the dose and the ideas for dosage regimen. Okay. Next, outcomes of phase one. So in phase one, we are going to see the safe dose range like we discussed, and we are going to see the bioavailability like Cmax, Dmax, AUC. Next, nature of ADRs. If there are any ADRs or tolerable doses which we have find out, we have to see the nature of ADR. Next, we'll to some extent we'll be getting the activity secondary phase objectives. Like your phase two objectives are like to see the activity and therapeutic benefits, right? So that is not our primary objective in phase one. But whenever we are giving certain dose ranging from 500 to 1000 grams of paracetamol, obviously the fever might be reduced or certain changes. So those kind of things will be seen at a minimum level in phase one, right? Yeah, this is same slide which we had kept previously. It one is done to see the safety, tolerability dose, peak and PD. So that's about your phase one and the challenges and advantages. So advantages is to see the safety dose of the drug, safety profile of the drug, and tolerable dose of the drug. And the disadvantages is ethical challenges and like to it's not that easy to get approval for doing phase one clinical trials for ethical issues or people involving as healthy one because we don't get much people in clinical trials much, right? And certain unexpected areas happen. So we should have a protocol how we are going to treat that unexpected areas. Okay. So yes, that's about your phase one. About phase two, we'll see in next class. Okay.